Greetings to judges, teachers, and fellow students. I am Samuel Pang, and together with Kelson Look and Keith Low, we form the team from St. Joseph's Institution. Our investigation will be on the effect of hydrogen peroxide imbibition on water spinach growth rate. To start off, water spinach is a leafy green vegetable which sees widespread consumption in the Southeast Asian region. So the imbibition of seeds in H2O2 solution has proven to be a promising method to increase the growth rate of plants in general. Hence, we can assess the viability of this method to improve the growth of this food source. As you can see on the photo on the left, H2O2 acts on water spinach seeds through a variety of mechanisms and actions. Firstly, we have the removal of pathogenic microbes. So they protect the seeds from disease-causing bacteria, viruses, etc. Secondly, we have the weakening of the endosperm. And H2O2 also stimulates the scarification of seed, which breaks down the seed coat and allows more efficient absorption of nutrients and oxygen and so on. After imbibition, H2O2 may also serve as a direct supply of oxygen as shown in the oxygen element in the compound, and this allows seeds to conduct aerobic respiration when it decomposes into oxygen and water. So what pushed us to pursue this investigation? Despite literature suggesting that H2O2 imbibition of seeds is bound to assist in the growth of water spinach, very few people have conducted this experiment on its effects specifically on water spinach. In addition, H2O2 is also a very strong oxidizing agent, and as shown in the previous slide, we have selective mRNA oxidation and protein oxidation, and this in high concentration could impair the plant growth. Therefore, we were motivated to find this ideal concentration to use for imbibition of water spinach seeds specifically, and this will be useful to boost harvest of the water spinach crops. So the hypothesis of our investigation is as shown below. This is because as the concentration of solution increases, its beneficial effects on the seed's growth become more pronounced, which is why the growth rate increases. But beyond a certain concentration, the cytotoxic effects of hydrogen peroxide would outweigh its benefits and instead lead to poorer growth rates. So this is the list of apparatus and chemicals in the table. We have undergone checks for safety hazards and precautions before choosing these items. So firstly, we diluted a 6% hydrogen peroxide solution with water to obtain 7 different concentrations of H2O2 solutions. These were then added into each of the 7 centrifuge tubes. After soaking for 24 hours, the seeds were then taken out and placed in each petri dish, which were labeled with the corresponding concentrations of the solution the seeds had been imbibed in. In the following 10 days, the respective root and shoot lengths of each seed was recorded at 24-hour intervals. And this experiment was repeated 3 times so that 3 sets of data could be collected and averaged. As for the measurements, since water spinach seeds undergo epigynous germination where the seed coats do not separate the shoot from root, we distinguish the root from the shoot by colour, root being the white parts and the shoots being the green parts. So this is the picture of my setup for the experiment. So now we will watch a time-lapse video over 10 days of the experiment to show the growth of the water spinach seeds. So this is how we achieved a quantifiable measurement of growth rate in cm per day. One thing we took note of during the calculation was that since water spinach is known to have poor probabilities of germination as shown in the very inconsistent graph on the left side, only the roots and shoot lengths of germinated seeds were used to calculate growth rate. This was to prevent the measured growth rate from being lower than a true value when less seeds germinated for certain concentrations, and vice versa. This precaution is necessary to increase accuracy of our data. Kelson will now cover on the results and discussion section of our investigation.
Here is the brief overview of the data we collected presented in various forms. Firstly, we tabulated the relationship between the length of seedlings against time, including the best fit line, to visualize the average growth rate of all the seedlings. From the findings, 0.15% concentration, indicated by the green lines, had the greatest growth rate of about 0.391 cm per day. The other concentrations that were lower than 0.15% had lower growth rates but did not dip below our control concentration of 0%. Those concentrations above 0.15% also began to decrease in growth rate, with the lowest being 0.3 in purple. Next, we tabulated the gradients of the best fit lines into tables to get a more precise look at our previous primary and process data. Lastly, we plotted the mean length of roots and shoots against all the 7 concentrations of hydrogen peroxides the seeds were soaked in onto a bar graph. It is evident that the growth of seedlings increases to an optimal concentration of 0.15% and starts to dip once concentration exceeds 0.15%. However, there is an anomaly in the seeds soaked in the 0.05% concentration, which will be justified by us later on. Overall, our data partially supports our hypothesis. For instance, the average growth rate of seedlings increases as concentration of hydrogen peroxide increases and peaks at an optimal point of 0.15%, with the exception of the growth rate of seedlings at 0.05% concentration. The scarification of seed coats by hydrogen peroxide could have been the main factor in the improvement of growth rates with the concentration, since the seed coats of water spinach seeds are highly impermeable to water and oxygen. Moving on, we have a possible explanation for the anomaly of growth rate at 0.05%, which is due to the limitation of measuring seed growth by length instead of mass. This is because lengths of plant shoots and roots fluctuate with temperature and soil moisture, conditions which are difficult to maintain. Lastly, there is a decrease in the average growth rate of seedlings. This is largely due to the cytosic effects of hydrogen peroxide that rise with increasing concentrations, impairing growth rate of seeds as hydrogen peroxide concentration exceeds 0.15%. This is shown in the lower growth rate of 0.3% concentration than our control concentration of 0%. In conclusion, since the trend observed in the results are largely as predicted in our hypothesis, it can be concluded that water spinach is affected by hydrogen peroxide imbibition in a similar manner to previous studied plant species.
Due to systematic errors in the methods used in measuring water spinach growth, a more robust approach to the study could be proposed. The length of roots and shoots may change due to uncontrollable factors, so they are not a definite indicator of the plant's growth. Additionally, it is difficult to accurately measure length of roots and shoots with a ruler, especially because they tend to curl and curve as they grow. Using high-precision electronic mass balancers will provide a more accurate assessment of growth rates by allowing the use of more conventional and well-founded methods, such as the calculation of relative growth rate. The accuracy of results can also be improved by conducting the experiment in a controlled environment. Since external conditions such as temperature and light intensity affect growth, this will ensure that the only variable affecting germination and growth of the seeds is the concentration of hydrogen peroxide used for treatment. One way to achieve this will be to construct an incubator where the temperature and light exposure to the water spinach seeds can be easily fixed to a certain value. The surrounding conditions of seeds during imbibition could also be studied further. Raising the temperature beyond room temperature will increase the rate of diffusion of hydrogen peroxide into the seeds, speeding up the imbibition process. However, raising the temperature might also damage the seeds, or cause the hydrogen peroxide to decompose too quickly to take effect, making this an interesting avenue of research. Lastly, other methods of applying hydrogen peroxide could be explored. For instance, dilute hydrogen peroxide solution could be used to water the plants instead of tap water. Since it has been established that exposing water spinach seeds to hydrogen peroxide increases growth rates, this could be another viable alternative of increasing crop yields using hydrogen peroxide. This section explains how our research is relevant to a chosen community. Since water spinach is crucial to populations in Southeast Asia, this region is a target community of our study. It is the largest global consumer of water spinach, and this vegetable can be found in the cuisines of cultures across the region. It is also a valuable source of nutrients, such as vitamins A and C and magnesium. Improving the growth rates of water spinach will help to ensure food security of the countries, which consume it as a keystone of their diet. Additionally, water spinach is also cultivated in Southeast Asia by countries such as Indonesia because it grows well in tropical climates. It is then shipped to other Southeast Asian countries like Thailand and Singapore in order to generate revenue for the country. Farming is a prevalent industry in these countries, so improving the crop yield of water spinach increases the revenue generated by agriculture and boosts the country's economic growth. Treatment of water spinach seeds with hydrogen peroxide is also especially applicable to farms in Southeast Asia, since many farms utilize traditional or manual means of farming. It only requires low concentrations of hydrogen peroxide and a large container to soak the seeds in, making it cheaper and much quicker than technology-intensive methods like genetic engineering. This makes treatment with hydrogen peroxide an accessible option for farms in Southeast Asia, and may be a viable way to improve the growth of water spinach. Lastly, hydrogen peroxide is highly biodegradable and leaves very little environmental impact.
making it an ideal agent to raise crops with. After being used in fields, it quickly decomposes into oxygen and water. These substances carry little to no harmful effects to ecosystems surrounding the farms. Compared to other methods of assisting plant growth, such as fertilizers which contain nitrates and ammonia, hydrogen peroxide is much more sustainable.
We thank you for your attention, and we hope that you have understood more about our investigation on the effect of H2O2 imbibition on water spinach growth rate.